Next topic is viral diseases. In viral diseases, first is common cold. Viral disease caused by rhinovirus. So common cold is caused by rhinovirus. They infect nose and respiratory passage but not the lungs. So common cold is caused by rhinovirus. It infects nose and respiratory passage but not the lungs. The infection remains only till respiratory passage. It cannot affect the lungs. Common cold. Symptoms: nasal congestion, nasal discharge, sore throat, hoarseness of voice, cough, tiredness, and headache. Common cold usually lasts for 3 to 7 days, and transmission is by inhaling the droplets resulting from cough or sneeze of an infected person directly or through contaminated objects such as pens, books, cups, doorknobs, computer keyboards, or mouse, etc. Both common cold and pneumonia are airborne diseases. So question can be asked like common cold and pneumonia are. So answer is airborne diseases. Next is AIDS. AIDS, the full form is asked very often. Acquired immunodeficiency syndrome. It means deficiency of immune system acquired during lifetime of person. It indicates that it is not a congenital disease. Syndrome means group of symptoms. AIDS was first reported in 1981. In last 25 years, it has killed more than 25 million people. AIDS is caused by HIV virus, that is full form is human immunodeficiency virus. HIV virus is a member of a group of viruses called retrovirus, having reverse transcriptase enzyme. So what are retroviruses? Those who have reverse transcriptase enzyme. HIV have ssRNA genome enclosed by an envelope known as capsid. So it has ssRNA genome enclosed by capsid. HIV is enveloped virus that contains two identical molecules of single-stranded RNA and two molecules of reverse transcriptase. It was asked in NEET 2016. So how transmission happens for HIV virus? Sexual contact with infected person. First is sexual contact with infected person. Second is by transfusion of contaminated blood and blood products. Third is by sharing infected needles, as in case of intravenous drug abusers, from infected mother. Fourth is from infected mother to her child through placenta. So transmission of AIDS is very very important as per theory point of view and MCQs both. AIDS is not spread by only touch or physical contact, it spreads through the body fluids. So answer is how AIDS spreads. It spreads by body fluids. When body fluids of the infected person are in contact with healthy person, transmission happens. People who are at higher risk getting this infection: persons having multiple sexual partners, drug addicts who take drugs intravenously, persons requiring repeated blood transfusions, and children born to an HIV infected mother. So there is always a time lag between the infection and appearance of AIDS symptoms, that is known as incubation period. First the infection happens and then the symptoms appear. So time lag between the infection and the appearance of symptoms is known as incubation period. Incubation period may vary from a few months to many years, that is usually 5 to 10 years. After infection, HIV enters in macrophages where RNA of virus replicates to form viral DNA with the help of enzyme reverse transcriptase. Viral DNA incorporated into host cell DNA and directs the infection cell to produce new virus particles. The macrophage continues to produce virus, therefore macrophages are called HIV factory. Simultaneously, HIV enters into helper T lymphocytes. So it is a very very important question regarding HIV. It enters into helper T lymphocytes, replicates and produces progeny viruses. The progeny virus is released into the blood, attacked other helper T lymphocytes. So HIV attacks helper T lymphocytes. Progeny viruses released in blood attack other helper T lymphocytes. This is repeated, leading to a progressive decrease in the number of helper T lymphocytes in the body of infected person. Just remember HIV, helper T lymphocytes. During this period, person suffers from bouts of fever, diarrhea, weight loss. Due to decrease in TH cells, person starts suffering from infections that could have been otherwise overcome, such as those due to bacteria, especially mycobacterium, virus, fungi, and even parasites like toxoplasma. The patient becomes so immunodeficient that she is unable to protect himself herself against these infections. Diagnosis test for AIDS is ELISA. Very, very important question. Diagnostic test for AIDS is ELISA. Full form of ELISA is again very, very important. This can be one multiple choice question, that diagnostic test of AIDS is what? It is ELISA. It is very, very important. Since long it is very, very important. So diagnostic test for AIDS is ELISA. Full form of ELISA is enzyme linked immunosorbent assay. I am repeating, enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay is full form of ELISA, and diagnostic test for AIDS is ELISA. Enzyme linked immunosorbent assay. Western blotting is used as confirmatory or supplemental test for AIDS. So first, diagnostic. This again is very, very important question, that confirmatory test. So diagnostic test is ELISA and confirmatory test is Western blot. Routine test is PCR. So you don't have to remember only test, you also have to remember that whether it is diagnostic test, whether it is confirmatory test, or whether it is routine test. So three types of tests are there. Diagnostic test for AIDS is ELISA. AIDS को डायग्नोस किस टेस्ट से कर सकते हैं? ELISA से. और कन्फर्मेशन कौन सा टेस्ट देगा? वेस्टर्न ब्लॉट. एंड रूटीन टेस्ट इज PCR.